Share the slides. Okay. Can you see things? Is it okay? Can you see the shared slides? Yeah. Okay. So we're talking about. Why is it? What? Yep. How do I get the pointer? Okay. So we're going to be talking about chapter 12 of Campbell's, which is mitosis in the cell cycle. And we're going to talk about the organization of genetic material first. So all of a person's genes or their genome is in the form of DNA, right? And it's stored in the nucleus. And humans have a lot of DNA. Like people have on average 3 billion base pairs worth of DNA. Like if you were to take a singular cell and put all of its DNA from end to end, you'd have a big line that's 2 meters long. I actually have like a demonstration set up for this. I have a tape measure. And that's 2 meters. It doesn't even fit in the screen. This thing's taller than I am. Right? So like my point is that like humans have a ton of DNA, and the way that they fit all this DNA inside a nucleus, which is like a really, really tiny little speck, is that they get the DNA inside the chromosomes and they wrap it around the histone proteins, which are these purplish ones that you can see here. And they wrap it twice around the histone protein, and they wrap it again and again and again. And that's how they condense a huge amount of DNA into just like little bits, right? And generally, when you hear the word chromosome, you can think of these X-shaped things, right? But on a daily basis, your genes look a lot more like this. It's like a big mass of blurry stuff that you can't really tell the X-shaped thing because the X-shaped thing only appears during cell division, like mitosis or meiosis. Because like when it condenses, it's easier to pull apart and to divide into different parts. Because if you have the big jumbled mess, you don't really know where you're pulling, right? So that's going to be our topic for today, which is mitosis. And the first thing I'm going to talk about is the cell cycle, in which there are two main parts. The first one is mitosis, which is the dividey part, and then there's interphase, which is the everything else part, right? So interphase, there not much is happening. It's just like normal cell things, and the cell is growing and developing, right? So it's accumulating nutrients in order to do this one. And G1, like the G in G1 and G2, can stand for either gap or growth, which was always kind of weird to me. Like, why not just choose one, right? So in G1, you accumulate nutrients in order to divide the DNA. And synthesis or S phase is when the DNA within the cell is replicated by DNA polymerase to make two copies, which is how you can have two sides. Like these two sides have the exact same DNA. They're exact replicas of each other. And that's what happens in the synthesis phase. And G2 phase prepares the cell for mitosis. It's like making sure everything is enough to do mitosis. And there's also this special phase called G0, which only happens like some of the time. Like the two main ways it happens is, for example, if you have DNA damage between G1 and S, this little line right here, that's a checkpoint. Like, oops, sorry. And if the cell in G1 wants to replicate, it has to make sure that its DNA is clean and not messed up. Because if it's messed up, then it can accumulate mutations and eventually turn into something bad like cancer, right? So if it finds DNA damage before replicating, the cell can go into G0 phase in order to isolate itself from damaging itself more. Or the cell reaches maturity and it doesn't really need to divide anymore. Like your neurons, for example, they divide, and when you have fully developed brain and nerves, it just stops growing. So that means after mitosis, it goes straight to G0 and it just doesn't do anything after that. Which is why, like, if you get brain damage, you can't really heal. Unlike like your hands, if you get scarred, that can heal, but like your brain can't heal after it gets hurt. And the other part, which is the main topic for today, is going to be the mitotic phase or M phase, where the cell actually divides. So now I'm going to talk about the different steps, phases in mitosis. The first one of which is going to be prophase, where the cell is doing final preparations for division. Like you can see the really nice two-stranded, almost X-shaped things right here. That's the chromosomes going from the jumbled mess into the nice X-shaped thing so it could get pulled apart. And the nucleolus, which is like the middle section where the RNA is synthesized, is also gone. You can see it's no longer there. And these things, which are called centrosomes, they are what pull the DNA apart physically. They divide and they move towards different ends of the cell. And you can see that in the next picture over here. So this is prometaphase, which is, it shouldn't really be a phase because it's just a continuation of prophase. But Campbell says it's a separate phase, so I guess it is a separate phase. So in here, you can see the difference between these little yellow things. This one's still moving, and here it's done moving, and it's on opposite sides. And the chromosomes are fully condensed now. You can see it's very short and very straight and parallel, I guess. And the nucleus is no longer there. So that's how these yellow things can connect to it. And these yellow things are what are called microtubules, right? They are one of the three kinds of cytoskeletal elements, and they project off of the centrosome to attach to the centromere, which is the center part here. In the center part of the chromosome is a centromere, and that's where the microtubules, like the yellow things, connect to in order to pull the chromosome apart. And they don't connect directly to the centromere. They connect to, you see in the photo, there's a little black speck. That's what is called a kinetochore, which is another type of protein that the microtubules can connect to. So these ones that do connect are called kinetochore microtubules because they connect to kinetochores. And those that don't are called non-kinetochore, right here. And these still do have functions, like they can push off of each other to lengthen the size of the cell so that there's more space to divide, I guess. And that's prometaphase. Oh. The next phase would be metaphase. And this one, I would say, is the simplest of them all, because it's just aligning all of the chromosomes along the middle. And the middle part is what is called a metaphase plate, right? So this plate is an imaginary line across the center of the cell. Like the cell is a sphere, and you cut it in half. That's where the metaphase plate is. It's not really a thing, more like an idea, right? So it lines up in the center so that when it gets pulled evenly instead of being pulled more on one side than to the other, which can cause problems. So that's metaphase. It's very simple. And anaphase is the actual divide part of mitosis, where you can see the yellow finger-like things are shrinking. And as they shrink or depolymerize, it pulls on the kinetochore, which are the little black dots, so that it creates pressure on the chromosome. And the chromosome is attached at the centromere by these other proteins called cohesins. And these cohesins are cut at the same time it's getting pulled. So there's no more adhesion between these chromosomes. So there's no more adhesion between the chromosomes, and it gets pulled apart, right? And during this phase as well, the non-kinetochore microtubules are pushing against each other in order to lengthen the cell, so there's more space to pull apart too. And the last phase of mitosis would be called telophase, where the cell is trying to reform itself, right? You can see all of the yellow lines are now gone. That's because the microtubules have depolymerized and turned back into singular tubulin molecules instead of this big long chain. And the chromosomes are no longer nice and X-shaped. They're beginning to look more strand-like and more like the jumbled mess from earlier. And you can see that the nucleus is reforming. Like I said earlier, when the nucleus dissolves in the parent cell, it doesn't just disappear, right? It forms vesicles that are stored for this step here. And when telophase begins, the vesicles formed off of the nucleus of the old cell come back and reform around the chromosomes. And that's how the nucleus is reestablished. And in this picture, you can also see the nucleolus that's forming. And there's a start of what is called cytokinesis. You can see the little V-shaped thing on the edges, and that's the cell trying to divide its cytoplasm. And you can see that on the next page here. So this process is called cleavage, and it's caused by a thing called the cleavage furrow. And this forms after telophase is complete in the center of the two cells, right? You can see it's forming like a little line. And the cleavage furrow is the nudge that's inside the cell and is formed by primarily two proteins, and that'd be actin and myosin. And you can remember actin, those are microfilaments that like structure the cell, and myosin, that's the protein in your muscles, right? So in your muscles, myosin pulls on the actin strands in order to contract or relax your muscles. And that's kind of what's happening here. Like the myosin is contracting and pulling the actin closer until it's really, really small and it pops out. Like I would imagine it's like a balloon and you stick your fingers in the middle and you try to strangle the balloon, except when it pops, the ends stick back together instead of just popping. And after cytokinesis is finished, the formation of the daughter cells is done because you would have one cell here and one cell here, and they don't no longer share any other materials because it's done replicating. And after that, the cell is finished here and can proceed to do it all over again. And you can see in this diagram here that this is actually like a really short phase. Like the entirety of mitosis can last like an hour at most, while interphase can last like a day. And that's because dividing is much simpler than growing. And the fastest step in mitosis is the anaphase. This can take 2 minutes, like 2 minutes to pull it apart. And that's like really fast, you know? It's quite cool. So one differentiation between animal and plant cells is that plant cells, they have cell walls, right? And you can't really just squeeze a cell wall for it to break apart because it's like a really, really solid material that doesn't break really easily, right? So instead of forming a contractile ring around about right here, it forms something called a cell plate, which is like a line along the metaphase plate. And over time, it forms new cellulose, and that forms a cell wall as well. So like you divide, but the cell wall of the parent cell doesn't change. You're just making a new cell wall between the two divided cells. So I guess it doesn't really become bigger. It just stays the same size. And I should have a video here, but it's not. Yep. So here's a quick video of a hamster cell doing mitosis. Hey, isn't that cool? Like you can see the whole cell divide. Yeah. And you can see around here, here, here. Hmm. Like the pulling of anaphase happens really, really fast, right? You can see the first 20 seconds of this video is just the chromosomes squirming around and forming the metaphase plate. and the last 10 seconds is it squirming around to form the new cells. And there's only like half a second in between where anaphase happens, because anaphase is like a really short phase. And I'm going to talk about bacteria now. Like bacteria don't actually do mitosis because they're really simple and they don't have organelles to share. So bacteria are basically just like a cell membrane coating with DNA inside it and like nothing else. So what they do is a process called binary fission. Am I in slideshow? I'm not in slideshow. So they do binary fission, right? Because they don't need to pull apart their chromosomes since they only have like one big one. So how this starts is that you have a cell that kind of looks like this one down here. And you have one origin of replication, which is where DNA replication begins. And as the chromosome is replicated, the origin of replication moved to opposite sides of the cell. Like you can see there's one here and there's one here, right? And after all of the DNA within the bacterial cell is multiplied, it gets cut right between to form the cleavage furrow again. And instead of using actin and myosin, they don't really have that. So they just have a tubulin-like protein. Like it's kind of similar, but it's not really the same thing. And it just pinches off. So there's no PMAT at all. It's just you divide and then you pinch. So there's S phase and there's cytokinesis. And that's because like bacteria are much simpler than animal cells, right? So while it takes a lot of effort to divide an animal cell, bacteria just kind of they divide really fast. Like E. coli cells can divide once every 20 minutes, as opposed to humans like about a day or so. That's because they're really simple. Yeah. And I think this is the last slide about mitosis. I think I'm going kind of fast. And this is on the regulation of the cell cycle. Like, if you don't have any regulation of the cell cycle, the cell can potentially just replicate and then replicate again and do it again until they form cancer. And that's just a tumor, and that's very bad for you. Yeah. And in order to not grow cancer cells, there are multiple different ways to limit the rate at which mitosis happens, right? So one of these ways is called an MPF, which is mitosis promoting factors. And these are formed when a CDK or cyclin-dependent kinases bind with something called a cyclin. And on its own, a CDK doesn't actually do anything at all. It's just like a molecule that's inside the cell. But when it binds with cyclins, it forms a mitosis promoting factor, which allows the cell to move into M phase, the mitotic phase. And without MPF, the cell either cannot or doesn't do it properly. And so after mitosis, the cyclin is degraded so that it doesn't replicate again unless the cell knows it's capable of doing so. Like if the nutrients aren't available for it, the cyclins aren't made, and MPF is not formed, which means mitosis does not happen. And this is like one of the big ways the cell regulates mitosis from happening. There are also checkpoints between like main phases of the cell cycle that prevents damaged DNA from being replicated and damaged cells from becoming duplicated. Like, for example, I have talked about the one earlier between G1 and S. There is a checkpoint that roots out all cells that have DNA damage. Come on. Oh, uh, Sorry, technical difficulties. Okay, where was I? Checkpoints. So the checkpoint here roots out anything that has DNA damage and set it to G0, right? And there's also a checkpoint here at G2 before the cell actually divides that checks again for like the amount of nutrients that the cell has to make sure it has enough to sustain the new two cells. And also checks for DNA damage again. And if there is DNA damage after the S phase, the cell essentially can't do much about it. So it does something called apoptosis, like you can see here. And what apoptosis is that the cell essentially just kills itself. It's cell-mediated suicide, right? And the cell kind of implodes on itself, and all of its nutrients and stuff is left to be cleaned up by macrophages and stuff. And so these checkpoints make sure that no bad DNA or harmful mutations are made into the next generation of cells where it can replicate and become tumors or cancer. And there are also one last form of regulation, which are signaling molecules or interactions. So growth factors, if you remember from last class, there are these things called GPCRs, where like if a growth factor binds to the protein, it can activate a transduction pathway that regulates gene expression, right? So one of these ways it can regulate gene expression is how fast the cell divides. Like if you remember, the Ras protein is a protein that promotes transcription factors and the division of DNA, which means if you have an activated Ras protein because of growth factors, then the cell will divide faster than normal. And this is usually good. Like it, for example, it can form your limbs or help you heal a wound. But if there's like a mutation there or an excess amount of growth factors, you can still form cancer cells. And there are two other kinds of interactions between cells that prevent them from dividing too fast. The first one is density dependent inhibition, which means that if there are too many cells compacted together, the cells there don't divide anymore. Like if you were to have a flask and you fill the bottom with like a solution that cells can grow on, and you add cells there, the cells there will divide until it forms like a flat layer, like only one singular layer of cells on top of that solution. And after that, it's not going to replicate anymore because after that, it's going to be like too much density for the cell to survive. Like, for example, if you take a little knife and you scrape off a part of that layer, the cells will reform that part that you scraped off, but it won't add any more to it, right? Because otherwise, you can have cancer. If the other one is called anchorage dependence, which means cells have to have something to grow on. Like, for example, the ECM, the extracellular matrix, is a matrix of proteins that a cell can attach to, or like your bones or something. Like, without something a cell can attach to, the cell will not divide. For example, if you got a test tube and you put cells inside a liquid and you swirl it around, the cells won't divide because there's nothing to grow on. Unless you let it settle and let it grow on the sides of the tube, it's not going to grow. So it cannot grow suspended inside a solution. And these two are two very important ways a cell can limit division. And they are also two of the biggest things that are not that don't get followed by cancer cells. Like for example, cancer cells can just grow and grow on top of each other to form tumors, and they can fly off and form different tumors elsewhere because they don't have to grow on something. And I think that's it for mitosis. Right? Yeah. Questions? Now how I can make a poll. Oops. Oh. How many people do we have in the meeting, Eric?
Right now, there's 23, but excluding us, 19, I think. Wait, no,
Okay.
23. 23, my bad.
So I'll wait until 15 people answer, I guess.
Yeah.
Okay. Yeah, okay, that's 13 is good enough. So the vast majority of you guys said E, which is the right answer. Should I share results? Share results. Yeah, okay. Oops. So A would be prophase. This one would be also prophase. This one's metaphase. And this one's also metaphase. So this is the only one that's actually anaphase, because anaphase is the one that divides, right? And here's another one. It's very blurry.
Wait, isn't that prometaphase?
What?
Isn't that prometaphase, not metaphase?
Prometaphase and metaphase are basically the same thing.
Oh, okay.
Okay, that's good. So 10 out of 16 of you guys said D, which is right, because this inhibitor is stopping stem cells in the middle of DNA replication. And this means that and DNA replication happens in S phase, right? So it's stopping the cell in the middle of DNA replication, which means it's stopping the cell in S phase. Yeah. And it, the next chapter I'll cover is meiosis, or Campbell's 13, I guess. So genes are meiosis is a process that allows for producing offspring, right? And the genes are inherited through gametes and not your body cells. And if you can remember, gametes are like sperm and egg cells, and somatic cells are everything else. And the reason why genes are inherited through gametes and not somatic cells is because, like, if you remember earlier, gametes are haploid or a single N, and somatic cells are diploid or 2N. And if you were to have genes that were inherited through somatic cells, you would double the amount of genes you have every generation, and that would be pretty bad. So that's why it's inherited through gametes. And there are two types of reproduction. The first one is asexual, where basically the organism's offspring are the exact same as the parent cell, which is like the binary fission example that was given earlier for mitosis. And sexual, which means that the offspring are genetically unique. And in order to make the offspring genetically unique, the cell has to create something that's different, right? It has to be different from the old chromosome. And that's what meiosis does. It creates gametes that are different from your normal somatic cells and it has more variety. And you can see here, there are two chromosomes, and these are called homologous chromosomes, right? These are in the X-shaped ones. So if you can see, the two sides are sister chromosomes, chromatids, which means that they just have finished DNA replication and are the exact same. They contain exact replicas of each other, right? They have the same genes and the same alleles. While these two are called are called homologous, which means that they don't have the same alleles, but they do have the same genes. Like they have different versions of the same gene at the same places. Like for example, if you can see a color that matches each other, for example, this blue one, they might both code for eyeball color, but one might code for brown eyeballs and the other one might code for blue eyeballs, right? So that's what it means for like loci. That's the spot on the gene. And you have 22 pairs of these and one pair of these. And sex chromosomes determine your sex, right? So if you have XY, then you are a male, and if you have XX, then you're a female. And sex chromosomes don't necessarily have homologous chromosomes because X and Y chromosomes are significantly different sizes. Like X chromosomes are like much bigger than like a Y chromosome. Like they're hugely different. And you can see in this little graph that there are two types. There's haploid and diploid, and gametes are haploid, and normal body cells are diploid. And in order to fertilize and form a new organism, the diploid multicellular organism has to go through meiosis, which is what we're going to be telling about, to form gametes, which can combine to make a diploid organism again. So meiosis, in a lot of ways, is very similar to mitosis, right? It's basically doing mitosis twice. So here, they divide it into meiosis 1 and meiosis 2, because it has to divide twice to form a haploid cell, right? So you first have your normal parent cell, which is a germ cell in this case, because germ cells create haploid cells that are gametes, and it replicates its chromosomes to form sister chromatids, right? And in meiosis 1, the difference between meiosis 1 and mitosis is that instead of splitting open sister chromatids, you're splitting open homologous chromosomes, right? So along the metaphase plate during meiosis 1, the homologous chromosomes match together to form something called a tetrad. It's basically like the two X-shaped chromosomes linked together, and they go through this process called crossing over. And this is like a really cool process. Like, for example, you can see here, they both have legs, right? And when they're layered on top of each other, they can create something called a chiasmata, which is where they touch, and they can swap their legs. This is also one of the ways that meiosis can create more genetic diversity to make sexual reproduction more random and create more diverse offspring. Because like when you swap the legs on the chromosomes are no longer the same alleles and genes as your old chromosomes. So it's something completely new. And during meiosis one, the homologous chromosomes are pulled apart instead of the sister chromatids. Right? Does anyone want to tell me why it's haploid after meiosis one? Like after the diploid cell divides once, it's already haploid. Why is it already haploid?
Because it goes through two divisions.
Is that louder?
Because it goes through two divisions.
No, it becomes diploid after only one division. And
Oh,
that's because like after the homologous chromosomes split, the homologous chromosomes are pulled into two different cells, right? Which means you have one cell that has like one side of homologous chrome. Yeah, Isaac?
Because like it only has like 23, like it only has like half the amount of chromosomes because the meiosis one basically kind of like took half of them from each.
What? I'm sorry, I didn't really understand that.
Like so the chromosom the two homologous chromosomes are like in different cells now.
Yeah, that's true. But how is it haploid?
Like because the homologous chromosomes only count for one out of the two normally diploid cells have.
Yeah, I think you get the idea. Yeah. So like after meiosis one, their homologous chromosomes are separated into the cells, which means there are only the X-shaped things that are in the cell. You see, there are only these X-shaped things, and if you can recall, these X-shaped two sides are called sister chromatids, and they are exact replicas of each other, which means you basically just have double the amount of a haploid cell. So because it's just a double of a haploid cell, this cell is already haploid, just with more DNA. And meiosis 2 is where these again align along the metaphase plate and is pulled apart again to form these little ones that only have half of these, which only have half of these. So they're haploid and only contain half of the DNA within a cell. So like if you were to merge two of these, you can form one of these, which is how fertilization works, I guess. And the next slide is about genetic diversity. And there are three, three forms of genetic diversity. That meiosis can do. The first one is independent assortment, which is also like kind of the thing that Mendel was talking about. But here it means that every time one of these, sorry, one of these chromosomes are pulled apart, the chance of them being pulled into the different cells is completely random. Like, for example, this one being pulled into two blues and two reds is the exact same of it being pulled into one blue, one red, and one blue and red, right? So because it divides twice, the chances of the 23 chromosomes ending up in separate places, like there's a huge array of different permutations that you can do with 23 chromosomes dividing twice. So that's like the combination of 23 different chromosomes is like a huge amount. And that creates a lot of genetic diversity, right? And I've already talked about crossing over, where they can exchange legs and make totally new or recombinant chromosomes. And lastly, random fertilization is that the sperm and the egg meeting is completely random as well. Like produce like a couple thousand sperm every time, and the chance that you choose one of those to meet the egg is really small. And like because of the number of permutations you can do with 23 chromosomes, like the one sperm that actually fertilizes the egg and the one egg that is actually produced, they have like there's a huge amount of different cell arrangements that you can make, which makes the offspring a lot more diverse. So there are no two people on Earth that have the exact same genes. Like I'm pretty sure the statistic was like you have to make like 33 trillion kids with the same two parents in order to have like clones of a person. That's like really cool too, because genetic diversity, you know? And I think we're on to problems again. Yeah, problems. We do a poll or oh, that's the poll. Is this still an old poll? Yeah, I think that's still the old poll. So this is kind of like the same question as earlier, right? Yeah, this is kind of the same question. I don't think you need to do this. It's just the same thing again. How do I make a poll? Eric, please help me.
Is it gone now?
Yeah, how do you make a poll, Eric?
Oh, let me make one real quick.
Can you make one for me? Thanks.
Oh, never mind.
Thanks. Take your time with this one. It's like a little bit more complicated. Hmm. You haven't responded, you can like try, you know? Doesn't matter if you're wrong. You get 15 or right. Yeah, I'd say 13 is good. Okay. So the answer for this one is E. Like most of you guys chose B, which is 1 and 3, which is wrong because like this one. So meiosis, you first like duplicate the DNA, right? That always happens first. So now you have double the amount of DNA, but then you divide once, so there's one little bump, and then you have to divide again, so there's two little bumps. So this is meiosis one. After meiosis one, you form the first meiosis product, and this is meiosis two, which forms the second meiosis product, which is haploid. So you can see there's, yeah, Arush?
From you like it's the same logic as like 2 and 4 have like the same logic, but doesn't it go back to like the like wouldn't 2 be correct as well?
Yeah, but it says respectively meiosis and then mitosis, and there's no option for 2 followed by 1.
Yeah, that's right. There's no
Oh,
two followed by one.
huh.
Although I would say the graph of two makes a little bit more sense than four.
Yeah, it does.
Because it goes back to only the same amount, right?
Mm-hmm.
It's a little weird, but like this still makes more sense because there's no other one that has one for mitosis.
Yeah.
So this part should be lower here, but other than that, it should be right. Like you duplicate, so you have double, divide once, you have the same, and divide again to have half. And mitosis is much simpler. You divide once, you replicate your DNA once, and then you divide to make the same amount. Yeah. Is there anything else in this? Why did you guys still choose B? 1 and 3. Yeah, the DNA does not replicate twice, so there's only supposed to be one bump here. And also, that's not mitosis. Oh, Uh, I'm done with my slides. We still have 10 minutes. What to do, Eric?
Should we do Usabo questions like last time?
Read useful questions like last time. Smart.
Just end off like 2018.
2018 Usabo. Was it opens or
I think so.
it should be this one, right? What could be the other one?
Oh, Michelle wants to do semi-final questions.
Oh, all right. We could do the semifinal one then. Okay. We didn't do this last time, right? So, all right. Can we pull again?
Dude, semis are scary. You sure you guys want to do semis?
Yes.
Don't do it. Don't do it. I'm cooked.
We haven't even explained this part yet.
Yeah, like there we have not taught like any of these.
Oh, wait a minute. We don't have the results for number one.
All right, we'll see how they do on this one, and then we can decide, right?
Oh, for independent research, I never made that. Wait, what question are we doing?
Number one. Okay, I think that's good enough. So most of you guys chose number one, some of you guys chose E, and one of you guys chose B. And correct me if I'm wrong here, but I'm pretty sure the answer is B. Okay. So, yeah?
Yeah.
Okay. So, progesterone that it forms the endometrial lining, right? So it maintains the lining in case pregnancy does happen. Chorionic gonadotropin, I'm pretty sure that's released after the baby is formed, so
Mhm.
it maintains the pregnancy. And estrogen, like secondary sex characteristics, I guess.
Yeah,
And
it's part of the, I mean, it definitely plays a role in the menstrual cycle, but it's not responsible for the surge.
and estrogen is not a real thing, so it has to be LH.
Yeah.
Should we do opens? Is it too hard?
I don't think, dude, I don't think you guys are ready for semis. Yeah. Semis is scary. Once you guys read more, you guys
Okay, we've done the
probably
first
tackle
two.
a few more. But right now I think opens practice is better.
Well, you can do this one. Number 11. Plants? Can we do plants?
We haven't done plans yet.
We haven't done plants yet. We could
Just follow.
gauge how well they do, right?
Hey, you should just give us all a challenge.
Ceilings growing, which have lots of fun.
Oh,
Oh, this one's fun, I think.
uh, Eric, how do I make a new poll?
Oh, wait.
Did it? Oh, I did
Yeah.
it. Good. Is the poll open? I don't have any answers.
Yeah, this question is a lot taken. We could give him a couple minutes. 2 minutes maybe.
I'm actually not really sure how this.
Let me take a look. Mutant. See these correct below. Loss of function are true. Numbers from stage. Mm.
That's about 2 to 3 minutes. We can reveal answers, right? Okay, so we have a tie between 2 and 4. Is it 5, Anthony?
Is it 5?
Is it E?
I think it's E.
Yeah, it's E. Um, this one's actually not that easy. It's pretty.
This is harder than the semis one.
I wouldn't say it's harder than the semis one. Maybe
Yeah.
that's a specific question, but semis is a whole different beast. This reads more like a semis question. I'm not gonna lie though. You're right. No, so first of all, the triple response is imagine you're a seed. You're deep in the soil. You just germinated. You're sending up your shoots to develop into a plant so you can photosynthesize. But as you're growing up, you hit a rock. Now you're not strong enough to break through the rock. So what do? Well, that's what the triple response is. The triple response is a way for plants to um, think of it as an algorithm that the plant uses to get away from upward obstacles. So step one, it stops vertical growth. So it will stop growing upwards. It will stop pushing up against the rock, right? So it won't damage its own tissues by like forcing itself into the rock. Second step is it grows thicker as so that's to give the shoot more support, right? Because you're damaging yourself a little bit as when you bump into the rock. You're trying to buffer yourself up. Third step is sideways growth. So you're gonna start growing sideways to like kind of just go around the rock. So that's like the dumb problem. Like I say dumb, but it's a clever problem solving method. Um, and so the reason that ethylene isn't involved is ethylene is the stress hormone. And when the plant um presses against the rock, the tissues start getting damaged. There's pressure, so ethylene, the stress hormone, gets released. Now, ethylene causes this triple response. Um, in this case, the question is saying, okay, you these plants oh, sorry. Loss of function mutation in the ethylene receptor.
So you
These
can't like
plants,
so like the ethylene doesn't work anymore, right? So it can't go through with the triple response.
yeah. I just saw that some of them were like small, got confused. These plants can't respond to ethylene because the first step in the pathway is cut off. There's no receptor. So that's number one. There's no other way for ethylene to exert its effect. It can't go into the second step of the pathway and attach itself there. It has to be the first one. So that's why it won't respond under any circumstances. The number part two, I don't think we can go through them all. But part three, mutant constitutive activity leads to inactivation of kinase. Um, this one just makes sense. Like even if you don't know that uh there's specifically a kinase in this pathway, it says that it leads to inactivation of kinase, which is important for the triple response. So if you inactivate the kinase that's important, then the response won't happen, which is what happening here, which is what's happening here. And five, if this mutant treated with this ethylene synthesis inhibitor, mutant will not respond. It doesn't matter what it's treated with. It won't respond to ethylene either way. And besides, this is the ethylene synthesis inhibitor, so this will be like a double negative. Except in this case, it doesn't become positive. So yeah, that's why the answer is E. Um, I would say I would ask if you guys have any questions, but we are at 8 o'clock. I guess if you have questions, hang around, you can ask. Otherwise, uh, peace. Enjoy the rest of your week.
Why?
Bye bye.
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 Bye bye.
Stop sharing, right? Okay.
Okay,
We always still have left.
let me kick this. Let me remove this Nico guy. Okay. Oh, wait, don't report. Okay, wait, Larry, that was a great lecture, not gonna lie. Um, I I like how you didn't read off the slides. I could tell you did prep. I could tell you knew what you were talking about. And you didn't yap as much as me, which is good. You moved through the content fast. I think you just did good. Like that's really what election needs. It's like prep and knowledge of the content. Uh yeah, I don't really have much else to say. Um In-person meeting next week. Grace and I will figure out a plan tomorrow and tell you guys about that. Probably just gonna be games, but we'll figure out which one specifically. I think Angela is going to get the food, um, for the meet. She says that she can get it the day of, so we don't need to worry about that. Um, and yeah, I'm pretty sure the Usabo registration is open. You guys can start signing up. Uh, we should send out information about that. Uh, I think we can like just send a Schoology post with like the link and here, let me find the link. Content
It's like the first link you sent, right?
for what'd you say?
Like the first link that you sent today, that's the registration link?
No, that's I just sent the Usabo website. Hold on,
Oh.
Usabo 2025 site, the registration. So it should be this one. Here. That one. So you just select the school and then you click, do the things. Yeah. Okay, let me check out the Schoology group bio. I'll send out a message or a thing on the Schoology group later. Anything else you guys want to talk about? Anything you want to?
Hmm.
No.
I don't think
Okay,
so.
okay. Then I'll see you guys sometime. Maybe tomorrow. I don't know, but I'll see you guys. All right. Bye bye.